Okay, so here are the answers to the questions at the end of the previous chapter. So why don't you pause the video while you tick them off, and you can carry on with the next chapter, which is reproduction. Now there are two forms of reproduction. One of them is sexual. Now, the definition of sexual reproduction is the joining or fusion of male and female gametes. Now, it's not enough to say male and female gametes meet or mix or come together. They have to fuse or join. You must say that in the exam. And it's also involving the mixing of genes from two parents. And it's not good enough to say the mixing of characteristics from two parents. It's the mixing of genes from two parents. And the outcome of sexual reproduction is that there is variety in the offspring. That's not the definition, but that's the outcome. And there's asexual reproduction. Definition is there's no joining or fusion of gametes, and there's no mixing of genes because there's only one parent. The outcome is there's no variety in offspring, and we describe the offspring as clones because they're genetically identical to the parent. Plants can be cloned. There are two methods. You can take cuttings, or there's tissue culture. Cuttings are very simple. You cut off a suitable piece of stem, you place it in compost, and you keep it moist. It'll grow roots and it'll grow into a new plant. And then there's tissue culture. Now the starting material in tissue culture is small groups of cells from the parent plant called explants. Now these can be grown in petri dishes into plantlets with the use of hormones, and then these plantlets are grown on into plants. Now this is carried out in huge polytunnels and greenhouses. It's a very large volume operation. You need specialist equipment, but you produce a lot of plants. Now both these techniques produce clones. Cuttings are very quick, very cheap. You don't need any special equipment for it. But tissue culture produces many more offspring from one plant. So what about cloning animals then? There are two methods of cloning animals. Firstly, there's embryo transplants. Now this involves splitting apart clumps of cells from an early embryo. So an embryo is a ball of cells. If you catch it early, before the cells start to specialize or differentiate, you can split apart that embryo into several clumps of cells. And these clumps will start to divide again as if from scratch. So each of these clumps of cells will then go on to grow into an embryo and then into a fetus. So these separated clumps of cells are then transplanted into the host mother's uterus. Uterus. Where else is it going to be transplanted? It's the uterus. You must say uterus. So this diagram shows embryo transplant. We start out with a fertilized egg, which carries out some cell divisions until it forms a ball of cells, which is an embryo. If you separate that embryo into several clumps of cells, then each of those clumps of cells will start to divide again as if from scratch, and each of those clumps of cells will then form an embryo. If these embryos are then implanted into a host mother's uterus, then it can go on to develop into a fetus. So each of these offspring is a clone of each other. The other method of cloning animals is adult cell cloning, or adult cell fusion, it's sometimes called. Now in this, we take an egg cell out of one animal and enucleate it, which means suck out and throw away the nucleus. We just want the empty egg cell. We can take a nucleus from an adult body cell from another animal. For example, we'll take a skin cell and we'll remove the nucleus, and it's the nucleus from this adult skin cell that we want. The nucleus is inserted into the empty egg cell. Now, an electric shock will stimulate that egg cell to start dividing. and after several cell divisions, an embryo is formed. This is a ball of cells, and at this stage it's inserted into the host mother's uterus. Again, you got to say the word uterus, to develop. So here's adult cell cloning. We have sheep A and sheep B. We take an egg cell out of sheep B. We suck out the nucleus and throw it away. We just want the egg cell. We'll take a skin cell from sheep A and we'll suck out the nucleus, but it's the nucleus that we want. So that nucleus is inserted into the empty egg cell. We treat it with an electric shock, and that stimulates it to divide. At the ball of cells stage, the embryo stage, it's implanted into the host mother, into the uterus, and the offspring is a clone of sheep A because that's where the nucleus came from. Now, genetic engineering is not the same as cloning. Cloning means making lots of identical offspring. Genetic engineering means altering the DNA of an individual. So, genes can be cut out from chromosomes using enzymes. And these genes can be transferred to other species. See these terms, cut out and enzymes are underlined. Those are the words to use in the exam. These genes can be transferred to the cells of other organisms at an early stage of development. And these other organisms then develop the desired characteristic that's coded for by that gene. So let's look more closely at what we're doing in genetic engineering. So we start out with our target gene. Now this gene codes for the characteristic we want to import into our crop plants or our livestock, our farm animals. And this gene will be found within some other species, some other organism. Maybe it's a bird that's got this gene that we really want, or maybe it's a bacteria, or maybe it's a fish or a worm. Anything that's got a gene that codes for a desirable characteristic can provide us with our target gene. And this gene is cut out using enzymes. And here's our host DNA. This belongs to the organism that we want to transform. The host DNA is cut using enzymes, and the target DNA, the target gene, is inserted, again using enzymes. So that modified DNA is put back into the host organism, so the host organism now codes for that characteristic, for example, for resistance to a disease. So plant crops treated in this way are called genetically modified or GM crops. And in this way, they can gain any characteristic that you want. An example is resistance to insect attacks or resistance to herbicides. GM crops usually show increased yields. That means we get more food from them. But some people are concerned about GM crops because they're not sure about the effect upon the ecosystem or effects upon human health if they enter the food chain. Okay, so that's the end of this chapter. Hopefully you've printed off all the notes so you can have a go at these questions now. You'll find the answers at the beginning of the next chapter. Good luck!